Assalamu alaikum wa rahmatullahi wa barakatuh. Dear students, this is the male reproductive system from module 207. The male reproductive system is formed of primary sex organ, which is the testis. It is mixed compound tubular gland, mixed as secrete hormones as testosterone, and exocrine as produce the sperm. The secondary sex organs are the ejaculatory ducts, which include the tubular recti, reed testis, vas efferentia, epididymis, vas deferens, and ejaculatory duct. Accessory glands to provide seminal fluid with nutrition, including the seminal vesicles and prostate, as well as the bulbourethral gland and the male organ. Here, dear students, is a testis stained with hematoxylin and eosine microscopic slide showing cut section in the seminiferous tubule. Seminiferous tubule. First of all, you know that the stroma of the testis covered with three coats: tunica vaginalis, albogenia, and tunica vasculosa. In each lobule, we have 250 lobule, 1 to 4 seminiferous tubule. The length is 30 to 70 centimeters. So they appear convoluted. Cut section appears rounded with lumen, lined with spermatogenic epithelium, lying on the basement membrane. It is stratified epithelium. The spermatogenic epithelium is formed of spermatogenic cell and cell known as Sertoli cell, which is a supporting cell. Okay, here I'm having a basement membrane of this seminiferous tubule, and this is basement membrane of the other, and so on. In between them, there is the interstitial tissue containing the interstitial cells of Leydig that secrete testosterone. In the section of the testis, in the seminiferous tubule, you should identify many cells. I'm going to tell you their names now. Okay, the spermatogonia, primary spermatocyte, secondary spermatocyte, spermatid, and spermatogonia, as well as the Sertoli cell. How could I differentiate between them? Okay, dear students, look here at that diagram. This is the basement membrane of the seminiferous tubule. And if I draw this, I'm going to continue the seminiferous tubule, and here is another. So this is the interstitial tissue. On the basement membrane lie cell known as spermatogonia. This spermatogonia divide by mitosis and differentiate into primary spermatocyte that enter in the first meiotic cell division and change it into secondary spermatocyte, then enter second meiotic cell division to be spermatid, then the spermatid undergo metamorphosis and change it into sperm. This cell is the Sertoli cell. I cannot see the boundaries because the Sertoli cell is a large, tall cell. Starts from the basement membrane and reach the lumen. This cell supports all spermatogenic cells. In the section, I can see the spermatogonia, primary spermatocyte. I cannot see the secondary spermatocyte. First meiotic cell division is I cannot see the secondary because it enters in the second meiotic and change it into spermatid so rapidly. So I cannot see the secondary spermatocyte. So I can see spermatogonia, primary spermatocyte, spermatid, and spermatozoa, as well as Sertoli cell. And I'm going to see another cell at the basement membrane known as the myoid cell that contract in order to let the cells move toward the lumen. How could I differentiate between them? Again, okay, Okay, dear students, how to differentiate between all these cells? The key is to put your eye on the basal lamina. If I'm having cell spindle nucleus like this, it is the myoid cell present on the outer side of the basal lamina. Okay? Then the cell that rests on the basal lamina is the spermatogonia. We have many types of spermatogonia. Dark type and pale type A, and we have type B. The type B undergo mitosis and proliferation change it into primary spermatocyte. Both of them have diploid number of chromosomes. The primary spermatocyte entered the first meiotic cell division and changed to secondary spermatocyte that rapidly changed into the spermatid by second meiotic cell division, so it's difficult to see secondary spermatocyte at this section. So, what cells can I see, doctor? First, the myoid cell, the spermatogonia, which rests on the basement membrane, the primary spermatocyte, large cell, and I can see clumps of chromosomes. The spermatid, we have two types of spermatids, early and late. The early is small, rounded. Okay, the late appears as if it's like going to change into sperm. Then the sperm at the lumen. Okay, I can see another type of cell. What this cell? The Sertoli cell. I cannot see boundaries of the Sertoli cell. Why? Because its supporting cell contain pockets at the lateral aspect protecting all these cells. Okay, doctor, again, this is a seminiferous tubule, and this is another one, and this is another one, cuts in the seminiferous tubule. How many cells I'm going to identify? 1 myoid cell, 2 spermatogonia, 3 Sertoli cell, 4 primary spermatocyte, 5 spermatid. Okay? The Sertoli cell, the nucleus, appears elongated and pale. In between the seminiferous tubule, there is interstitial cell of Leydig. It appears vacuolated as it's rich in lipid droplets, which is testosterone. Then, dear students, the sperms produced by the testes will move at the male duct, which are intratesticular, known as tubulorectary testes, extratesticular as Ephranthes ductus, and epididymis, vas deferens, ejaculatory, and male urethra. I'm going to study what? I'm going to study the epididymis. I'm going to study the epididymis. It is a tall tube, 4 to 6 meters, 4 to 6 meters, compact in 7 to 5 centimeters or 7.5 centimeter, present with the testis. So under the microscope, the epididymis appears with the testis, anatomically related. It's formed of head, body, and tail. When I cut section here, in the lumen there are many sperms. Why? One of the function of the epididymis is storage of the sperms. The lining epithelium is pseudostratified, columnar, ciliated with long non-motile cilia, which is the stereocilia, long microvilli. Then a smooth muscle, then surrounded with smooth muscle. Loose connective tissue and smooth muscle. Cuts in the epididymis. Again, what is this, dear students? It is the epididymis, from where you know many sperms. The nuclei not more than 2 pseudostratified with long microvilli or the stereocilia, connective tissue and smooth muscle, very small smooth muscle, epididymis. We have the vas deferens. Then we have the vas deferens. The tail of the epididymis gives rise to the vas deferens. It's about 45 centimeters it ascends upward against gravity. So the muscle is very large muscle, thick muscle. Okay? The lumen is not folded, lined with pseudostratified, columnar ciliated. The upper part loose the cilia. Pseudostratified columnar ciliated at the lower half only, while the upper become non-ciliated. Then connective tissue, then the muscle layer. Very strong muscle. Inner, outer, longitudinal, and very thick middle, circular. Then we have the advantage. Please, dear students, concentrate here at this slide. Small rounded structure with lumen. We said we have the ureter, the vas deferens, which also present in the spermatic cord, and the fallopian tube. We have 3 criteria. 1, look at the lining epithelium. 2, look at the folds. 3, look at the muscle. Okay? In the ureter, the lining epithelium, transitional, vas deferens, pseudostratified, fallopian tube, simple columnar. The more folded in the fallopian, less in the ureter, and not folded in the vas. While the muscle, maximum at the vas, moderate at the ureter, very thin in the fallopian tube. Here, dear students, is the spermatic cord formed by vas deferens, pampiniform plexus of veins, testicular artery, and the most important component is the cremasteric muscle, which is skeletal involuntary muscle. Skeletal involuntary muscle. Again, there is spermatic cord. I can see the vas deferens, pampiniform plexus of vein, testicular artery, and cremasteric muscle. From the glandular tissue of the male genital system, we're going to study the prostate. It's formed of stroma and parenchyma. The stroma capsule, trabeculae, and reticular tissue. Here, the trabeculae of the prostate. The trabeculae of the prostate is fibromuscular. The parenchyma is formed of prostatic acini and duct. The prostatic acini arranged in 3 levels, arranged in 3 concentric layers: inner or mucosal, intermediate or submucosal, and the main prostatic acini or the peripheral component. The acini, these are the acini, and this is the connective tissue, which is fibromuscular, and here is the prostatic urethra. The prostatic urethra lined by transitional epithelium. Again, here's the prostate. What is this? The prostatic urethra, which is lined by transitional epithelium. And here is the fibromuscular connective tissue. And these are the acini. High power of the acini, I can see here the acini. The prostatic acini lined with simple columnar or pseudostratified columnar epithelium. The epithelium is affected by the testosterone level. When the testosterone level is low, it becomes reduced into simple cuboidal cells. In the lumen of the acini, I can see corpora amylitia, which is prostatic concretions. Concentric lamellae of calcified body frequently seen, increase with age. Again, this is the prostatic acini and this is the fibromuscular stroma. The last section in the male genital system is TS section in the penis. This is structure covered by skin. We said we have 4 structures in our curriculum covered with skin: the lip and the eyelid, the penis, and the mammary gland. Okay, as you see here, cut section of the penis. Here is the skin, then connective tissue, then tunica albuginea, thick tunica albuginea, and 2, corpus cavernosa, corpora cavernosa, which is an erectile tissue, interconnected vascular space lined with endothelial cells separated by smooth muscle collagen and elastic fiber, corpora cavernosa. And I'm having corpus spongiosum, surrounded with more elastic fiber, and the tunica albuginea is thin. The tunica albuginea here is thin. So here, tunica albuginea, as if it's eyeglasses, frame of the eyeglasses, and these are the corpora cavernosa, which are erectile tissue formed of blood vessels lined with endothelium, separated by smooth muscle, elastic, and collagen fibers. You can see here the corpus spongiosum, the same as the erectile tissue, but here the tunica here is very thin. Why? Because here is the benign urethra. Here is the benign urethra. Okay? Again, 1, corpus cavernosa or corpora cavernosa, 2 of them. Number 2, corpus spongiosum. Number 3, the benign urethra. Number 4, tunica albuginea. And number 5 is the skin. Again, corpora cavernosa, 1, 2, corpus spongiosum, 3, tunica albuginea, 4, the benign urethra. Now we're going to take the trial exam. Okay, what is this? In one word, it's testis, showing seminiferous tubule. How to identify the cells? The doctor told us, look at the basal lamina. The nuclei present at the basal lamina flat is that of the myoid cell. So E is myoid cell. A cell with elongated oval nucleus pale with prominent nucleoli, it is Sertoli cell. Please, dear students, okay, I'm going to show you this Sertoli, and this, and this, and this are Sertoli cells. Then the cell resting at the basement membrane, the spermatogonia, the spermatogonia. This cell that contain chromosomes, large cell containing chromosomes, B, primary spermatocyte. So A is Sertoli cell, B, primary spermatocyte, C, spermatid, D, sperms or spermatozoa, E, myoid cell. Again, dear students, this is testis, seminiferous tubule. Okay, how could I know the cells? Look at the basement membrane. Okay, A, cell resting on the basement membrane from inside, spermatogonia. B, large cell containing chromosomes, primary spermatocyte. C, elongated cell with the nucleus is elongated, pale with prominent nucleoli. All of these are Sertoli cells. Sertoli cells. Okay, I can show you. Okay, I can write there, for example, S. Okay, here Sertoli. Okay, here Sertoli. Okay, here Sertoli, here Sertoli. Okay, primary spermatocyte A, excuse me, spermatogonia A, spermatogonia A. This is also spermatogonia. Primary spermatocyte, large cell containing chromosomes, large cell containing chromosomes, which is letter B. Number C, number C is the Sertoli cell. Number C is Sertoli cell. Number D, spermatid. Number G, interstitial cells of Leydig between the seminiferous tubule. Okay? And name the tissue that contains the G is interstitial tissue containing interstitial cells of Leydig. Okay, dear students? And F is the sperms. This is an important, here I can see all cells. In this section, please confirm that you know all the cells. Okay, I'm going to revise them again. Standing on the basement membrane from inside A, the spermatogonia. From outside appears flat nuclei, E, myoid cell. Resting on the basement membrane from inside spermatogonia, from outside myoid. Large cell contains clumps of chromosomes, which is B, primary spermatocyte. Cell in which the nucleus appears elongated, oval, vesicular with prominent nucleoli, Sertoli cell, C. D, small cell, darkly stained nuclei, D, spermatid. F, sperms or spermatozoa, near the lumen, head and tail. Between the seminiferous tubule, there is interstitial tissue containing interstitial cells of Leydig, G. Okay? Here, dear students, okay, this is the seminiferous tubule, and this is another one. So this is the interstitial tissue. How could I start? One is the myoid cell. Two is the primary spermatocyte, many chromosomes. Three is the spermatid. Four is the Leydig cell. Five is the Sertoli cell. Again, this is the basement membrane. Any flat nuclei for myoid cell. Cell containing chromosomes, primary spermatocyte. The spermatid is small and rounded. 3. The Leydig in between the seminiferous tubule. 4. The Sertoli cell, elongated oval nuclei. 5. Sertoli cell. Okay? Dear students, this is Sertoli cell. Okay? Any elongated oval, it is the Sertoli cell. Okay? Here, dear students, is interstitial tissue. Here it is interstitial tissue. Look here, dear students. This is interstitial tissue. So the yellow arrow is the myoid cell. The black arrow containing chromosomes, primary spermatocyte. The green arrow, interstitial cells of Leydig. Identify the organ, it is testis. Here is the epididymis. One, pseudostratified columnar ciliated with stereocilia or long microvilli. Number 2 is the stereocilia. Number 3, sperms. Number 4, connective tissue and smooth muscle. Small round structure with lumen, we have 4 structures. Ureter, vas deferens, spermatic cord, and fallopian tube. What I'm going to do, look at the lumen. The lumen is not folded. Number 2, the lining epithelium, not more than 2 layers, pseudostratified. Number C is the muscle, which is huge as it pumps the sperms from downwards upward against gravity. Number D is the adventitia. So A, the lumen, B, epithelium, C, muscle, D, the adventitia. Vas deferens. Here, dear students, the spermatic cord. A, vas deferens. B, cremasteric muscle. C, pampiniform plexus of veins. Here is a very nice question. Mention one visible feature for A. Feature for A, any visible feature, you can say very thick musculosa. In the exam, the word visible feature is something you know, you saw at the slide from where you know it. Okay? That's the meaning of visible feature. Here, dear students, is the concretions present at the prostatic acini. This is the prostate. I can see the black arrow, which is the prostatic acini. The star is the fibromuscular stroma. The red arrow is the prostatic concretion called corpora amylicia. Here, dear students, is the prostate. Number 1, the prostatic urethra. Number 2, the mucosal acini. 3, submucosal for male. The lining epithelium of 1 is transitional epithelium. Here, dear students, is the penis. Okay, here is the tunica albuginea. 1 is the corpora cavernosa. 2, corpus spongiosum. 3, tunica albuginea. 4, benile urethra. Again, 1, corpora cavernosa. 2, corpus spongiosum. 3, tunica albuginea. And 4, benile urethra. Then we are going to study the labeled diagram. Here, dear students, in the testis, I can see the seminiferous tubule. This cell from the basement membrane to the lumen is the Sertoli cell with the elongated oval nucleus pale with prominent nucleoli and different types of spermatogenic cells, which are spermatogonia, primary spermatocyte, secondary spermatocyte, spermatids, and sperm in the lumen. In between the seminiferous tubule, there are interstitial cells of Leydig. Dear students, this spermatic cord, I can see the vas deferens, lined with pseudostratified columnar ciliated, and here with the connective tissue corium, then the muscle layer, inner, outer, longitudinal, and mid-circular. Then we have the adventitia. Here, pampiniform plexus of veins, testicular artery, and you cannot say that it is spermatic cord without the cremasteric muscle, which is skeletal involuntary. The last section here is the prostate. I can see here the prostatic urethra lined with transitional epithelium and the prostatic acini mucosal, submucosal, and main. I can see here the corpora amylicia. I can see the fibromuscular capsule and trabecle. Thank you, dear students. We have finished the male genital system in the module 207. Assalamu alaikum wa rahmatullahi wa barakatuh.